All right, let's talk about a revolution that's happening right now. But it's not in a lab with test tubes and beakers, it's happening inside a computer. We're talking about a completely new kind of AI that's not just, recognizing cats and photos. It's actually learning the fundamental rules of biology itself. And that's allowing us to build, to test, and to understand our own bodies in ways that were pure science fiction just a few years back. So here's how we're going to break this down. First, we'll build our AI toolkit. We got to understand the core tech that makes all this possible. Then we're going to see those tools in action, creating virtual tissues, discovering new drugs at frankly impossible speeds, and mapping the crazy complex universe inside ourselves. And then we'll look at what this all means for the future of medicine. So let's just kick things off with a massive question. What if we could test millions of potential new medicines not over, say, a decade, but in a single day? I know, it sounds completely impossible, but this isn't some sci-fi what-if anymore. This is a problem that scientists are solving right now with AI, and we're about to see exactly how they're doing it. Okay, let's get into it. Before we can build virtual organs or find brand new drugs, we need to understand the tools of the trade, right? So in this first part, we're going to quickly unpack the 3 key AI concepts that are the engine behind everything we're about to see. First up is something called contrastive learning. Imagine trying to teach a computer what a dog is without ever actually saying, hey, this is a dog. Instead, you just show it thousands and thousands of pictures. Well, contrastive learning is the AI that figures it out all by itself. It's kind of like it's playing a giant game of spot the difference to learn the essential features of, well, anything. And here's the really cool part, how it actually works. When the AI sees different pictures of the same dog, it learns to pull them closer together in its virtual brain. But when it sees a dog and, say, a chair, it pushes them really far apart. And by doing that millions of times, it builds this rich internal map of what makes a dog, all without a single human label. Our second tool is the diffusion model. Now, you have definitely seen this technology in action. It's behind a lot of the crazy AI image generators out there. The best way to think about it is like an artist who starts with a block of marble, or in this case, a screen full of static, and knows exactly how to chip away all that noise to reveal a perfect sculpture underneath. And this image shows you exactly how it does that. It's a 2-step dance. In the forward path, you take a perfectly clear image and just add noise, bit by bit, until it's total static. But the real magic, the part the AI learns, is the reverse path. It painstakingly figures out how to undo that damage, step by step. And once it masters that process, it can start with pure noise and create something entirely new and totally realistic from scratch. And our last tool is the Graph Neural Network, or GNN. Most data fits pretty neatly into a spreadsheet with rows and columns, but biology's messy. It's a tangled web of connections and relationships. GNNs are built specifically for this mess. Just think of it like a social network, but for molecules, where the AI doesn't just look at each molecule by itself, it understands its entire network of friends and connections. So the crucial idea here is something called message passing. See how each dot, each node in the network, is basically sending messages to all of its direct neighbors? By gathering up all those little messages, a single node gets a complete picture of its local environment. That's how the AI learns context. And in biology, context is everything. All right, toolkit's ready. We've got our 3 key AI concepts down. So now for the fun part. Let's see how they come together in our first incredible application, creating living virtual tissues that help us understand disease in a whole new way. So why is this so hard to do normally? Well, looking at real tissue is incredibly messy. Every single lab uses slightly different methods, every patient's tissue is unique, and the data you get back is unbelievably dense. And all that means that the insights you get from one study almost never apply to another one. It's a huge bottleneck in research. And this is where a model called VERT2s comes in. Okay, I know this diagram looks super complicated, but the core idea is actually really elegant. The AI looks at an image of tissue at every possible level all at once, from individual proteins at the top, to the cells they make up, down to the neighborhoods those cells live in, and finally the entire tissue sample. It learns how all of these layers connect and influence each other in one big unified model. And here's where it gets absolutely mind-blowing. Because the AI learns the fundamental rules of how tissue is put together, like which cells like to be neighbors, or which proteins always show up together. It doesn't just see what's there. It starts to understand what should be there. It develops a kind of biological intuition. And, just look at this. This is the proof right here.
In these examples, the scientists basically took a digital eraser to parts of the tissue image, hiding them completely from the AI. And on the right, that's the AI's reconstruction. It has literally filled in the blanks, redrawing entire cells and structures with stunning accuracy, just based on its understanding of what was around them. It's like it's dreaming the missing pieces of the tissue. It's incredible.
Okay, so from seeing inside our tissues to designing the cures, let's now take our AI toolkit and apply it to one of the single biggest challenges in all of medicine, finding new drugs. You see, the traditional way we find new drugs is painstakingly slow, it's astronomically expensive, and more often than not, it just fails. There are thousands of proteins in our bodies that could be targets to treat diseases, but we've only ever explored a tiny fraction of them because we just don't have the time or the resources. So here's the AI approach. And again, let's focus on the big idea. Remember contrastive learning from our toolkit? Well, here, it's used to teach an AI the physics of how a drug molecule, scientists call it a ligand, fits into the little pocket of a target protein. It learns what a good fit looks like versus a bad one. And once it understands that, it can screen a massive digital library of potential drugs to find the best matches with unbelievable speed. And the results are, well, they're just on another level. Using this method, researchers screened 500 million, that's billion with a B, compounds. They performed 10 trillion evaluations. And out of that, they found 2 million potential new drug candidates. And they did all of this on a single computer, in a single day. That is the insane power of AI at scale. Okay, so we can build virtual tissue, and we can find new drugs. But what about understanding the fundamental map of how our body even works? Now, we're moving from what proteins are to what they actually do in their natural habitat, the cell. The big problem with older AI models is that they were totally context-free. They'd give you one generic description for a protein, and it didn't matter if that protein was in a brain cell or a skin cell. But this new model, PINNACLE, is context-aware. It understands that a protein's job can change dramatically depending on its cellular neighborhood. And how does it do this? You guessed it, with our old friend, the graph neural network. PINNACLE builds this enormous interconnected map, what they call a metagraph, that links proteins to the specific cell types they live in and then links those cells to the tissues they form. It's learning the entire biological hierarchy, and that's what gives it that all-important context. So, let's zoom all the way out. What does this all add up to? Well, we are witnessing the birth of a brand new field, in silico biology. That's just a fancy way of saying biology that's conducted entirely inside a computer, driven by AI. Today, we've seen how this new era is letting us build virtual digital twin tissues, screen billions of drugs in a day, create these context-aware maps of our proteins, and even predict how our cells will respond to different treatments. We're really moving from just observing biology to engineering it. And that brings us to a final, pretty profound thought. For decades, we've talked about DNA as being the code of life. But now, with AI tools like these, we're not just reading that code anymore, we are learning how to write it. So the question we're left with is, if biology is becoming code,